Bueno, a continuación quiero invitar al podio al doctor Carl-Philipp Heisenberg. Él es alemán de origen, trabaja en Austria, en el Instituto de Ciencia y Tecnología de Austria, y nos va a hablar cómo el concepto de organización se aplica a eventos del desarrollo temprano utilizando modelo de Pez Cebra. Entonces invitamos a Carl-Philipp a dar su presentación.
Thank you, Miguel, for inviting me and allowing me to tell you a little bit about what we are interested in and how that relates to self-organization in development of organisms and organismal development. I have to apologize that I'm not speaking Spanish, so it has to be in English. And please, the questions are going to be at the end, I think, when we are sitting here. Good. So what I would like to speak about is the sort of history of self-organization, how it has been arisen in developmental biology. And one of the first experiments has been done on this phenomenon of self-organization in early embryo development, has been done on amphibian embryos. And just put here one example of an amphibian Xenopus leavis, a frog, and the different stages of Xenopus leavis development. Do we have a pointer, by the way? Probably not. No. If we don't have a pointer, I can just sort of point out with my finger where we are roughly. This is the egg, the fertilized egg, and then you're going through a process which is called gustulation, when the different germ layers are being formed in the embryo. The first tissues the embryo is forming, ectoderm, mesoderm, and endoderm. And from these germ layers, the entire embryo is being formed, and then eventually the entire adult organism arises. And the experiment I would like to tell you about is nearly 100 years old. And that has been done by two embryologists, Towns and Holfrieder. And what they used is they used an early stage Xenopus embryo, or amphibian embryo in this case. And they did sort of an ingenious experiment. They took out the epidermis, which is sort of the skin of the embryo, later on giving rise to epidermis structures in the embryo. And they took out another structure, which is the neural plate. And this is the tissue from which the nervous system is going to emerge. And then they dissociated the epidermis, and they dissociated the neural plate into the single cells. So essentially, everything was disrupted. You have now a bunch, an aggregate of intermixed cells, randomly intermixed cells, and then they were just waiting and not doing anything. And what happened then is that eventually out of this random aggregate of epidermal and neural plate cells, something developed which looks like I'm very good. Something developed which looks a bit like an embryo again. So you start with this sort of completely intermixed aggregate of cells, and autonomously, miraculously, somehow an embryonic structure evolves where the new ectoderm is going to be here, the neural plate is going to be here, and the epidermis is surrounding it. And that was sort of unexpected, because this is not embryo development. It is something which happens in a culture dish. And it somehow refers to the autonomous property of these cells to reform the tissue they are coming from. They have somehow sort of a memory what they have been before, and they are recapitulating this memory now and forming the embryo again. If this is, it depends a little bit on your definition of self-organization. If you, in a very strict definition of self-organization, you would say you start something from a completely homogeneous cluster of cells. In these cases, these cells are not completely homogeneous because they are coming from different tissues. They already have different fates, and you could argue this is not self-organization, but it is self-assembly and genetically encoded self-assembly of cells. But this is a fine line, and I'm happy to discuss it later when we are sitting in front. Now, what Holfrieder and Towns were doing, they were saying this is presumably some sort of affinity of these cells, and the dark cells, the epidermis cells, like to stick to epidermis cells, and neural plate cells like to stick to neural plate cells, and then they are sorting out and forming this pseudo-embryo. The idea of this cell segregation and tissue formation was then taken up by Malcolm Steinberg a few years later, 10, 20 years later, He was an embryologist and developmental biologist in Princeton in the USA. And what he wanted to conceptualize what is happening in these self-organization of tissues. And he proposed that tissues do behave like immiscible liquids. Like in this case, what I put in the picture here would be oil and vinegar, or oil and water. And if you intermix them, then you know, and everyone has seen that they are not intermixing, but that in this case, the vinegar is being surrounded by the oil, because the vinegar, the water, has a very, very high surface tension and wants to round up, and the surface tension is higher than oil, and then oil surrounds essentially the vinegar. So the idea is now that these tissues do behave like liquids, and when you intermix oil and vinegar, then they will sort out eventually, and you will have your clutch of vinegar, and you will have your bubble of oil, and then they are segregated from each other. Now, this was sort of an idea, and what then Steinberg did is he used tissues from different parts of the embryo, whatever embryo he used in this case, and he designed a little machine where he can put the tissue between two plates, and then he can deform the tissue, and by knowing about the force he is applying on deforming the tissue and the tissue geometry, he can derive the surface tension of these different tissues. And then he would assume if these tissues have different surface tension and he puts them together, then they will arrange in an interesting way. And this is just one example out of many examples he produced in his professional life. He took, for instance, here lymph tissue and pigmented epithelial tissue, heart, liver, and retina. These are different tissues from the embryo, and they all have different values of surface tension, with higher values, high surface tension, and lower values than lower surface tension. And in the example here of lymph and pigmented epithelium, the tissue which is green is the lymph bud. It has a higher surface tension. It goes in the middle because it's higher surface tension, and it would be the water, and it would be surrounded by the tissue with a lower surface tension, which would be the oil. So in principle, the idea that surface tension of tissues is informative about their behavior, the segregation of cells, was sort of then also first experimentally tested by taking now not only the tissues and putting them adjacent to each other and see which tissue engulfs the other tissue, but then repeating these experiments from Hohlfrieder and Towns, and in this case we have ectoderm, mesoderm, and then intermixing these two populations of cells and see if they can sort out again. And depending on the surface tension of the tissue they're going to form, one tissue will envelop the other tissue. And you can put that in theoretical models where you assume that you start with something where you have many small bubbles of tissues, which then these droplets of, let's say, oil or vinegar in a sea of oil, they will fuse these droplets, and then you will go from an intermixed population of cells into some smaller droplets which fuse together, and then you have one big droplet, which would be here, the vinegar being surrounded by the oil. Now we sort of used this concept of differential surface tension to see if that actually, because that all was shown to, in principle, ex vivo by taking out tissues or taking out cells and see what they can do, if that might be involved in embryogenesis. Really. Is this concept of tissues with different surface tension doing something relative to each other, is that relevant to development? And the organism we are using are zebrafish embryos, and very much like amphibian embryos, you go through a series of embryonic stages here, gastrulation, germ layers are forming, and then you're ending up here with the adult organism. Zebrafish is very useful because you can keep many different strains of zebrafish, transgenic strains, mutant strains in the laboratory, and this is just showing you a picture of a fish facility as we have it, where you can keep essentially thousands of fish and do experiments on them. I put in this movie here, which has been recorded many years ago, and it just shows you the first hours in the life of a fish embryo, and I think it's just nice to see. If it would run. Here we go. So you're starting with a couple of cleavage divisions which lead to the formation of the first embryonic tissue, which is up here. And then you will see that out of this blastoderm tissue, something develops which looks like a proper embryo. It takes a little while here. I'm not sure why that looks like that. And on the right side, you see the fish embryo emerging with a head and a tail down there. And now you can sort of appreciate that this looks a little bit like an embryo already, starting from, again, from a clutch of seemingly uniform cells. Now, I put in a few stills of this movie and just showing you in the schematics down there what is happening, just to introduce you into the process we would like to understand, maybe would like to feed in the surface tension idea. And you start here on the left side. That's quite sensitive to, I'm not sure what I'm doing here. Yeah, well, it's, you start here on the left side, you start with this undifferentiated cluster of cells, and then this cell layer, this clutch of cells is spreading over the yolk cell. The yolk is the brown mass here, and then you have gustulation when the red arrow, these are the cells which are going to the inside. They're segregating out from the blue arrow, which is up there, and they're forming two distinct layers or two different tissues. And the question we ask is essentially, could that be driven by differences in surface tension between these two populations of cells, ectoderm and mesoderm? The way we did it, first we did these tissue tension measurements now, taking intact embryos, and I'll show you how that looks like. So you can squeeze these embryos, you can look at the deformation, you have the force acting on these plates, you can measure the surface tension of different tissues, and then you can do the classic thing. Once you know the surface tension, you look into the embryo and see again how these cells are moving in. You have one population of cells which is moving in and becomes different from the other population of cells. You have the surface tension of these different populations. And then you can do the classical Townsend-Holfrieder experiment, the segregation experiment here on the left. Let's see. Here we go. Okay, in this movie, unfortunately, which should be the quintessence of my talk, is not working. Yeah, okay. So they should sort out, and you should see the red cells in the middle and the green cells surrounding the red cells. I'm not sure why this movie is actually not working. So this was just an example how you can look at self-organization and what the fundamental principles are of self-organization. They are inherent features of cells, their affinity to bind to each other, the ability to form tissues with different surface tension, which drives then a specific morphogenetic process, which is segregation of cells, engulfment of one tissue by another tissue. Yeah, so I'm pretty much at the end, and I thank you for your attention.